I'm really excited to introduce our presenters for today. So we have Gabriele Nocchi and Samuel Yeaman. And just a quick note too that we will also have Vashti Dunham from Weyerhaeuser join us for the Q&A portion following the presentation. And I will introduce her fully once we get to the Q&A. So before I hand things over to Gabriele and Samuel, I'll just share a little bit about their backgrounds. So Dr. Gabriele Nocchi is a bioinformatician and postdoctoral researcher in ecology and evolution. His research uses empirical genomic data to study evolutionary processes and to address ecological questions with a primary focus on forest trees. He earned his PhD at Queen Mary University of London in 2022, working in the group of Professor Richard Buggs through a joint collaboration with the Royal Botanic Gardens. And during his PhD, he worked on the genomics of British oak species and Asian white birch. And following his PhD, Gabriele joined Professor Samuel Yeaman at the University of Calgary as a postdoctoral researcher. And his projects there focused on repeated adaptive processes in plants, the genetic basis of local adaptation and hybridization in spruce, and the genomic basis of heat tolerance in canola. He is also involved in the REP-ADAPT working group, which is an international research consortium of over 100 researchers led by Professor Sam Yeaman. And then Dr. Sam Yeaman works at University of Calgary as a professor in the Department of Biological Sciences. He did his undergraduate degree at Trent University and his PhD at UBC with Mike Whitlock, as well as postdoctoral positions at the University of Neuchâtel and UBC. His research uses a combination of comparative genomics, individual-based simulations, and observational studies of population genomic variation in the wild to understand how evolution shapes adaptive traits. And current projects focus on studying the genetic basis of local adaptation in conifers and heat tolerance in canola, and also leading the REP-ADAPT working group studying the genomics of signature adaptation processes across hundreds of plants, animals, and fungi. So with that, I will pass it over to both of you today. Over to you, Gabriele and Sam.
Thanks very much, Rachel.
Thank you.
Thank you all for coming to listen to our talk. I'll start off giving a bit of an introduction, and then I'll pass it off to Gabriele, who will tell you in detail about what he's been doing. And from the kind of work that Rachel was describing that we do, you may kind of wonder what the interface is with trees. It all sounds a little abstract. I've been interested in using trees to understand evolution and evolution to understand trees for a long time, because there's such a depth of work looking at provenance trials in trees, and we know so much about how trees adapt to their environment that it really gives us a good foundation for all sorts of things. And it's nice to learn something that's useful for forestry. So we're going to talk a bit about this project on looking at the genomic basis of adaptation in the White and Engelmann spruce hybrid zone. And so just to situate this, I thought I'd talk about some of the kind of big picture issues that might be interesting and that this might inform. So if you could do the next slide. So, thinking about how we can actually use genomics to learn about traits that are valuable for forestry, I think there's a lot of open questions here. We have really a lot to learn in terms of what's encoded in the genome and how that drives things that matter for forestry. We might care about adaptation to abiotic stress, things like drought or heat or cold tolerance. And with spruce, we're really interested in understanding how hybrid ancestry, the proportion of white spruce versus Engelmann spruce ancestry that a given individual has, how does that change how they grow and how they're able to tolerate stresses? We're also really interested in understanding whether some regions of the genome, some genes, are particularly important for survival in Alberta. So do we see parts of the genome spreading throughout the range of both white and Engelmann spruce because they're advantageous to the climates around here? And then thinking about whether there's trade-offs and whether stress tolerance somehow, if we improve stress tolerance, do we decrease wood production, or is there no such trade-off? Can we actually select for both traits at the same time? So these are the kind of big picture questions we'd like to learn about over time. And we're really starting, in this case, looking in more detail at this hybrid zone as a way to get a window into how evolution shapes these traits in spruce. So next slide. There's been a lot of work done already on spruce genomics. So on the left-hand side, you can see a plot from John Degner's PhD thesis. He worked with Sally Aitken at UBC. And so this is showing the percentage of white spruce versus Engelmann spruce ancestry. And you can see through most of northern Alberta, we've got mostly white spruce, and then the southern BC coming up to the north. We've got Engelmann spruce and this kind of wide hybrid zone through central BC plateau, and then also a hybrid zone through the Rocky Mountains on the continental divide there. And the climate modeling has suggested that climate change is going to result in some losses of spruce in the parkland and foothills regions of Alberta. And so understanding how hybrid zone plays out in terms of climate change, I think, is a really important question. Next slide. There's been a lot of work done looking at the effect of artificial selections. This is from Ian McLaughlin's thesis work, also in Sally Aitken's lab, where he looked at the performance of natural versus selected seed lots in terms of their final height on the left, and then the percentage of cold injury on the right. And in particular for spruce here, he didn't really find any big effect of the gains that came from selection, from artificial selection on cold injury. So you can see from the black bars on the right-hand side, we're not seeing consistently higher cold injury among the selected lines, which is what we want to see. That's good. But this is also certainly a question that we need to understand in depth as we continue doing artificial selection. Next slide. There's some kind of interesting and surprising and kind of puzzling results that come from looking at the genome, where we can look across these big samples of populations from all across BC and Alberta and quantify how much of the ancestry of a given individual, so a given tree on this plot here, the triangle plot, is a dot, and then the proportion of ancestry, whether it's Engelmann spruce on the left-hand side versus white spruce towards on the right-hand side towards the one, gives us an idea of how much ancestry each individual has. You see the next slide. And what's interesting is that when we look at the effect of artificial selection, we can see that it's been favoring white spruce ancestry in most places. So we've been seeing increases in white spruce ancestry, but surprisingly, this doesn't really square with other results we've got, where we see that Engelmann spruce individuals tend to be a bit larger or higher overall. And so if selection is favoring height growth, why is it the case that we're also seeming to select for white spruce ancestry? And so there's a lot to understand in terms of how our artificial selection, our breeding programs, interact with this spruce hybrid zone. And that's kind of the motivation for what we're doing in this talk. So I'll pass it off to Gabriele now, and he can tell you a bit more about the details of this project that he was doing, and then I'll follow up at the end.
So hi, everyone, and thanks for having me here today. All right, so yeah, we'll be talking in a little bit more details about the project that I did with Sam while doing my postdoc in Calgary. So the main objective of our study was to compare the genomic patterns of local adaptation and hybridization between two replicated regions of the white and Engelmann spruce hybrid zone characterized by different geographies. So we had a very broad latitudinal zone spanning thousands of kilometers across Western Canada and characterized by gradual environmental variation, which is the one that you can see here in this figure. And then we had a second hybrid zone, a narrow elevational zone spanning only 100 kilometers in the Canadian Rocky Mountains and characterized by very steep environmental variation. And this hybrid zone was located here within the red square that you can see in this figure, and we will see a little bit more details in later slides. So those are the main research questions that we tried to address with the study. So first, we were interested in whether local adaptation is able to maintain species divergence across fine-scale environmental variation within the narrow elevational hybrid zone. So essentially, our study uses fine-grained sampling in the narrow elevational hybrid zone as a natural laboratory. So in the narrow elevational zone, we sample three very contrasting environments, which you can see illustrated here in this figure, which are located at very close geographical proximities. And we thought this could generate some fine-grained structure in the spruce individuals. The second question that we tried to address was how similar are the genomic patterns of divergence, environmental association, and directional introgression between these two very different geographies of hybridization, so elevation versus latitudinal gradient. And at last, are there some regions of the genomes which are particularly important for their role in adaptation, in environmental adaptation? So this is the study system that I'm pretty sure everyone here is well aware. So it's White and Engelmann Spruce and their hybrids, which altogether are also known as the interior spruce complex. So Engelmann Spruce on one side has got low tolerance of high temperatures and drought, is able to withstand shorter growing season and higher snowfall, sets buds earlier than White Spruce, and reaches higher elevation. On the other hand, we have White Spruce, which grows under more variable conditions, is able to tolerate both low winter temperatures as well as summer drought. However, it is restricted to valleys and lower elevations. So to study the broad latitudinal hybrid zone, which I presented in my first slide, we actually use this data set, which is derived from one of Sam's previous studies. So this is a huge data set with 249 spruce populations scattered throughout BC and Alberta. It included almost 600 individuals. It was sampled with between 2 and 3 individuals for most of these populations. So here, what we did, we performed DNA extraction from all these samples and performed DNA sequencing, and we ended up with roughly 260,000 SNPs after filtering. So what are SNPs? SNPs are single nucleotide polymorphisms. They are the type of genetic markers that we have been using throughout this study. So in very short, for those who don't know what is a SNP is essentially a position in the DNA sequence where there is variation between individuals. Essentially is one of the things that contribute to the diversity that you see between individuals. So what we did here, you already saw these results in one of Sam's slides. We perform an ancestry inference. So an ancestry inference is essentially a method which uses analyze the SNP patterns of the individuals to assign them to groups. In this case, the groups are the 2 species clusters, so White Spruce and Engelmann Spruce. So this map represents essentially results of our ancestry inference, where each of these pi is one of the 249 populations, and the color of this pi represents the ancestry composition of that population, with the gray color representing White Spruce ancestry and the green color representing Engelmann Spruce ancestry. So as you probably know, as you can see here, White Spruce is predominant in Alberta and reaches farther north than Engelmann Spruce, which instead is predominant in southern British Columbia. And then we have the hybrid zone that extend pretty much throughout central British Columbia, as well as along the southern Alberta and British Columbia border. So next, we also add narrow elevational hybrid zone. And for this one, we put together this additional data set called elevational data set. So here we sample 7 locations along the Trans-Canada Highway going through Canmore, Banff, and Yeo. As I mentioned before, we sampled 3 populations for each of these locations. So a valley population, a south-facing high-altitude population, and a north-facing high-altitude population. It was a total of 331 individuals with between 15 and 16 individuals per population. So here, as I mentioned at the beginning, in the narrow elevational zone, we sampled these 3 very contrasting environments located at close geographical proximity. So at each of the 7 locations that we sampled, these 3 environments were never separated by more than 10 kilometers. So those are very contrasting environments. Valleys, of course, are warmer and drier than either slopes. And then we have north-facing slopes that compared to south-facing are much cooler and wetter and harsher environment, as you can see here in this figure, are shaded throughout. For most of the days, they experience much less direct sunlight compared to south-facing slopes. So we thought that this environmental variation could generate some fine-grained population structure in our individuals. So here we use the same method. We perform DNA sequencing using the same approach, and we ended up with roughly 120,000 SNP markers. We perform ancestry inference again, and in this data set, the patterns are actually more interesting. We found 2 patterns of ancestry variation. So the first one is actually the expected pattern. So we do see the ancestry is varying loosely from almost predominantly Engelmann spruce in the northwest to predominantly white spruce in the southeast. And this is essentially mirroring the overall species distribution in this area. If you remember the previous slide, we have Engelmann spruce predominant on the British Columbia side, whereas white spruce is predominant in Alberta, and this is mirroring this overall distribution. However, what we also see here is the ancestry distribution is strongly reflecting the environmental context. So instead of just seeing this gradual shift from northwest to southeast with similar ancestry between the environments at each location, what we actually see, we do see some fine-grained structure within each location. So if you look at each of these locations, the ancestry composition is changing as we move from north-facing slopes to south-facing slopes and then to valleys. And this is explained much better by this next slide, which essentially has the same information of the previous slide, but just with a different representation. So here I'm showing the distribution of individuals per location and per environment. We have the 7 locations here in the columns, and the environments are in the rows. And each individual was classified as a category according to their genetics. And the categories were either pure white spruce or pure Engelmann spruce, either an Engelmann spruce dominant hybrid or a white spruce dominant hybrid, or a balanced hybrid, which is an hybrid close to a 50-50 ratio. And this is just to show this fine-grained structure that we found at location. So if, for example, you look at location 1 and 2, which are located in the part of our sampling range where Engelmann spruce is predominant, you see the north-facing slopes as well as south-facing slopes are dominated by pure Engelmann spruce as well as Engelmann dominant hybrids. But once we get into the valleys, particularly here at location 1, we see that the pure Engelmann spruce completely disappear and give space to Engelmann dominant hybrids. This shift is much more dramatic and pronounced on the opposite end of the distribution range. Here we see location 6 and 7, white spruce ancestry and white spruce dominant hybrids are dominating the valleys and south-facing slopes. But once we move on to the north-facing slopes, we actually lose all the pure white spruce, all the white spruce dominant hybrids, and there is this dramatic reversal towards Engelmann ancestry with mostly Engelmann dominant hybrids and also some pure Engelmann spruce. Whereas for the locations here located more in the middle of our sampling range, this shift seems to occur on the south-facing slope that show more of a mixture of ancestries. So beside assessing ancestry, what we were interested in doing in this study was to perform a genome-environment association analysis to identify SNP markers associated with the environment. So very briefly, most of these SNP markers are actually neutral. However, some are under selection. Are under selection because they might provide an adaptive advantage or improve fitness in certain environments. So essentially, those are the loci, the SNPs that we are interested in finding with this GEA analysis. So we are trying to find SNPs associated with the environment by looking at extreme patterns of SNPs across space. So to do this, we selected some environmental variables that we thought were pretty good at differentiating the niche of the parental species. So those are the variables that we use. Aspect, of course, which is the compass direction that a surface is facing. Slope, which refers to the steepness of a landform. Air temperature at 2 meters, which is pretty self-explanatory. Then we use 2 variables related to light availability. The first one related to indirect light availability, while the second one related to direct light availability. And then we use snow melt day, which is simply the calendar day of snow melt. And this variable was particularly good at discriminating the 3 environments that we sampled in the narrow elevational dataset. So for our genome environment association analysis, we actually didn't use these variables in the raw form, but we combined them using this method called redundancy analysis, which aimed to identify the linear combination of these environmental variables that explain the greatest variation in hybrid index, essentially the greatest variation in ancestry. So we did this separately for the two data sets, the latitudinal and the elevational. In both, we were able to explain a good chunk of the ancestry variation using these predictors. In the latitudinal data set, the most important variable was slope, followed by PV out, which is the variable related to direct light availability. Whereas in the elevational data set, the most important variable was snow melt day, followed by PV out. So I'm not going to go too much into details on the methodology behind this, but we perform our genome environment association analysis independently in each of the two data sets. And what we get at the end for each of our SNP markers, we get a p-value. So the lower the p-value, the stronger is the evidence of association of that SNP with the environment. So we did this analysis independently, and then we compared our results. So in this slide here, I'm just showing results from chromosome 1 of Spruce. So here this is what is generally called as a Manhattan plot. Each point here in this figure are our SNP markers located on chromosome 1. On the x-axis, we have the approximate position along the chromosome. The y-axis is the minus log 10 of the p-value. So essentially, the higher this y-axis, the stronger is the evidence of selection, the evidence of association of a SNP with the environment. And we have results for the latitudinal analysis at the bottom and the elevation at the top. So what's really clear by just looking at this is that there is a high level of repeatability. So most of the peaks are shared. So this is, of course, just the first chromosome of the 12 chromosome of white spruce, but the situation is actually the same throughout all of the chromosome of the species. And you can just see by eyes, by looking at the next few slides, you can see most of the strongest peaks are repeated between the data sets. And this is the case for all of the 12 chromosomes. Of course, we actually checked the numbers, and we found a very high correlation between the p-values resulting from the two genome environment association analysis, with not significant difference between the chromosomes. So next, we actually explored what we found with our genome environment association analysis. And for this, we focused on two categories of candidates. So the one in blue here, which I circle as an example, are the candidates that were identified in both analyses. So here in this set, we found many genes that were previously reported for their central role in plant growth and environmental adaptation, including many that were also already reported in similar studies in conifers. Whereas the other category of candidates that we focused on I just circled some example here, are the one in red. Essentially, those are the candidates that were significant in one of the two analyses, but not in the other. And this for us was interesting, as it could pinpoint genes or genetic loci that might be involved in the response to environmental factors specific to latitude, such as, for example, photoperiod and seasonality, or vice versa, regulating factors more specific to elevational changes, such as oxygen levels, UV radiation, temperature, or snow level, for example. So the last analysis that we did with this data set was what we call a genomic line analysis. So this was aimed at identifying whether there are some SNPs under strong directional introgression. So what does that mean? It means that these SNPs are moving from one species into the hybrids and then into the other species through hybridization and repeated backcrossing. And why they do so? Generally because they have an adaptive role due to their adaptive relevance. So with this analysis, we wanted to check whether we could first find SNPs under directional introgression, second, whether these overlap with the adaptive loci that we found in our genome environment association analysis, and once again, whether these patterns are repeated between the elevational and latitudinal transects. So we performed this genomic line analysis, and all you really need to know as an output, you get an alpha value, which can be either positive or negative. Essentially, the higher the absolute value of alpha, the stronger is the evidence of a SNP being under directional introgression. So once again, we perform this analysis separately, and we check the correlation of the resulting alpha values, and we found a significant correlation of directional introgression patterns. Of course, this was not nearly as strong as the correlation that we found between the GEA p-values, but nevertheless, it was significant and positive. So here I circle as an example, for example, these SNPs here are SNPs that were classified as positive alpha outliers in both the latitudinal and elevational analysis. And essentially, these are loci where the white spruce allele is favored everywhere, as the white spruce allele is moving into the hybrids and then onto Engelmann spruce. And with everywhere, we mean across both latitude and elevation, as they were candidate in both of the analyses. Whereas here you have the opposite case. So those are loci, are SNPs that were identified as negative alpha outliers across both latitude and elevation, and therefore are loci where the Engelmann spruce allele is favored everywhere. So in the next few slides, I'm just showing the Manhattan plots from our genome environment association analysis, chromosome by chromosome, once again. So those are the same results that I showed before, with the addition that I highlighted as triangles, our candidates for directional introgression. So the gray triangle are the loci where the white spruce allele is favored, whereas the green triangles are the loci, the SNPs, where the Engelmann spruce allele is favored. And these slides are just really to show you first that the candidates for directional introgression are located on some of our strongest peaks for environmental association, and also to show you the amount of repeatability between latitude and elevation, as well the amount of non-repeatability. So I'll just scroll through the next 12 slides just to give you a brief outline of what we found. So this is chromosome 2. You have a very clear example of repeatability here towards the end of the chromosome, and also some non-repeatability around here. Little bit of repeatability over here. Strong repeatability on this peak here. Some repeatability here at the beginning of this chromosome. So overall, we performed 4 analyses, 2 genome environment association analysis and 2 genomic clients analysis. In total, we found 638 genes. Many were actually previously reported for their contribution to adaptation in plants. We then found 18 candidate genes, which were identified by all 4 analyses, and these constitute our strongest set of candidates. So we are pretty confident that these genes have an adaptive role, as they were consistently retrieved by each and every one of our analyses. Then, as I mentioned in one of my previous slides, we also found 110 genes potentially involved in the response to environmental factors specific to latitude, such as photoperiod and seasonalities. And those are those ones here, as well as 3 genes potentially involved in regulating factors more specific to elevational changes, which are those one here, which are the candidates specific to our elevational analysis. So overall, this brings us to the overall conclusions of this study. So we found very strong evidence for local adaptation, even at fine scale, across a single valley, with a big effect of aspect and elevation, as we saw in the narrow elevational hybrid zone. As we move at each location, the ancestry composition changes as we move from north-facing slopes to south-facing slope and to valleys. Then we also found a small number of genomic regions that appear to be strongly beneficial across both white and Engelmann spruce that may be good for breeding, and those are definitely the 18 candidate genes that were identified in common between all of the analyses. And at last, we found broadly similar patterns of adaptation and hybridization between the latitudinal and elevational transects, which suggests that essentially natural selection is playing a very major role in shaping these genomic patterns of hybridization between white and Engelmann spruce. So this concludes the presentation of the study. I'll give the word back to Sam to give you an outline of what's in the plan for the future.
Great, thanks, Gabriele. So yeah, I mean, as Gabriele implied, we're still continuing lots of work on this. And one thing we've been doing is deepening the sampling. So the red sites here are places we've already been to and gone to get extra samples, extending this hybrid zone study. And then Ian McLaughlin has been doing a lot of work for the forestry community, sampling from the foothills regions, getting sampling as well. And so we're really interested in seeing at a broader level what would happen as we kind of cast the net more widely, and trying to look into developing some cheaper ways of doing genotyping. The genotyping we used here was expensive. It was about $160 per sample, which is way prohibitive for any kind of application. And so I'm working with some people at UBC to try to develop cheaper ways of doing genotyping that might actually be deployable on a broader basis. And one of the things I'm really interested in working on is trying to develop drone-based phenotyping. So actually using drones and spectral imaging cameras, we can actually do a pretty good job at estimating a lot of traits that are relevant for stress tolerance. So things like drought tolerance, and even trying to estimate hybrid index for a given tree. And so further work that we're planning in the future is just trying to get at taking these seeds and from this hybrid zone and really looking in common gardens and in provenance trials, how do they perform? Are there any trade-offs with growth and these climate tolerance traits that we think are shaping the adaptation in this hybrid zone, really to try to deepen our understanding there. And one thing I'd love to get from the forestry community is any examples of natural mortality events. If you see spruce dying off, we'd love to hear about it, because we're particularly interested in trying to see if we can use this genomics to understand and predict which individuals might survive versus die when there's a high mortality event. So if you see spruce dying off in the parkland area, for example, or anywhere, we'd love to hear about it. Next slide. Some of the other work that we've been doing that was that Vashti helped fund was doing a draft genome assembly for white spruce to improve this, and we've done this now. The technical details aren't that interesting, but the nice thing to see is that when you do a genome assembly, you want to have your genome put together into pieces that are the same size as the chromosomes, and we've gone a long way towards that. Not quite there, but our new assembly is 600 times longer than the previous one. So it's we're really developing these genomic resources to help us towards this goal. So one more slide, Gabriele. I think that's it. Yeah. So thanks very much, everybody. Thanks, Gabriele, for going over this in such detail, and we're really happy to talk in more detail about any questions.
Awesome. Thank you, both Gabriele and Sam. Thank you so much for walking us through your research and sharing all the insights that you found throughout the way. So yeah, really appreciate your time this afternoon.